Welcome to today's episode titled Dentition in Mammals. This topic is presented by Mr. K. Nagaraju, lecturer in Zoology. In this episode, we will explore the types, structure, origin, evolution, and significance of teeth in mammals, meaning of teeth and dentition. Teeth are hard, pointed structures present in the buccal cavity. They are attached to the jaw bones. Teeth occur in both fetal and adult mammals. Dentition refers to number of teeth, structure of teeth, types of teeth, arrangement of teeth. Teeth are found in fishes, amphibians, reptiles, and ancestral birds. Teeth are most highly specialized in mammals. In mammals, teeth are present on premaxillae, maxillae, and dentaries. Teeth are never present on palatal bones. Teeth show differentiation in shape and function. Functions of teeth. Teeth help in grasping and holding food or prey. Teeth cut, tear, chew, and grind food. Teeth act as weapons for offense and defense. Significance of dentition. Dentition is important for mammalian classification. Dentition helps in taxonomic studies. Number of teeth indicates approximate age of the animal. Dentition gives clues about food habits. Dentition helps in tracing ancestry and evolution. Toothless mammals. Not all mammals possess teeth. Completely toothless mammals, Echidna, Tachyglossus, spiny anteater, Myrmecophaga, true anteater. Secondary toothlessness. Platypus has embryonic teeth only. Adult platypus possesses horny epidermal plates. Whalebone whales have fetal teeth only. Baleen plates replace teeth before or after birth. Many edentates lose teeth in adult stage. Differentiation of teeth based on shape. Homodont dentition. All teeth are similar in shape and size. Teeth perform similar functions. Seen mainly in non-mammalian vertebrates. Among mammals, homodont condition occurs in toothed whales, dolphins, porpoises, armadillos. Number of teeth ranges from 2 to 200. Heterodont dentition. Teeth differ in shape, size, and function. This is the typical mammalian condition. 4 types of teeth are present: incisors, canines, premolars, molars. Differentiation depends on nature of food, method of feeding, attachment of teeth to jaw bones, acrodont, teeth attached to crest of jawbone. Teeth have no roots. Teeth break easily and are replaced. Found in fishes, amphibians, and reptiles. Pleurodont, teeth attached to inner side of jawbone. Tooth touches bone with side of root. Teeth are rootless. Found in Urodilus and lizards, Thecodont. Teeth embedded in bony sockets called alveoli. Teeth possess well-developed roots. Blood vessels and nerves enter pulp cavity. This condition is characteristic of mammals. Succession of teeth, polyphyodont. Teeth replaced continuously throughout life. Seen in lower vertebrates, not found in mammals. Diphyodont. 2 sets of teeth are formed. First set, called milk, deciduous or lectile teeth. Molars are absent. Erupt after birth. Second set, called permanent teeth, replace milk teeth. Remain throughout life. In guinea pigs and bats, milk teeth shed before birth. Monophyodont. Only one set of teeth develops. Seen in platypus sirenians, toothed whales, marsupials, some rodents and moles. Types of teeth in mammals: incisors, located at front of jaws, present on premaxilla in upper jaw, present on dentaries in lower jaw. Characteristics: single-rooted, monocuspid, sharp-edged. Functions: cutting, biting, cropping, food. Special cases: sloths, incisors absent. Rodents: chisel-shaped, open-rooted incisors. Elephant tusks, modified incisors. Lemurs, comb-like incisors for grooming. Canines, located behind incisors. one canine in each half of jaw, long, pointed, single-rooted. Functions: piercing, tearing, holding prey. Variations: carnivores, large and spear-shaped. Walrus, upper canines form tusks. Rodents and lagomorphs, canines absent. Absence creates a gap called diastema. Cheek teeth, premolars and molars located behind canines, used for grinding and chewing. Premolars, usually two roots and two cusps. Present in milk dentition. Molars, more than two roots, several cusps. No milk predecessors. Types of cheek teeth based on cusps. Triconodon, three cusps arranged linearly, found in fossil mammals. Trituberculate, three cusps arranged triangularly, found in fossil mammals. Bunodont, rounded cusps, seen in man and monkey. Secodont, sharp cutting cusps, seen in carnivores. Selenodont, crescent-shaped ridges, seen in herbivores like cow and horse. Lophodont cusps form ridges called lofts, seen in elephants and manatees. Modification of teeth based on diet: herbivores. Broad grinding molars. Canines reduced or absent. Upper incisors absent in ruminants. Carnivores, large sharp canines. Carnassial teeth present. Omnivores. Bunodont cheek teeth. Mixed diet. Aquatic mammals. Pinnipeds, conical teeth to hold prey. Toothed whales. Homodont teeth. Baleen whales. Baleen plates for filtration. Dental formula. Dental formula expresses number of teeth. Written for one half of jaw. Upper jaw teeth written above line. Lower jaw teeth written below line. Primitive Eutherian formula: 3, 1, 4, 3, 3, 1, 4, 3 times 2 equals 44. Teeth number constant within species. Useful in mammalian classification. Structure of teeth in mammals. General structure of a tooth. Structure of teeth is similar in all vertebrates. A typical mammalian tooth shows high specialization. Each tooth is differentiated into three main parts: crown, neck, root. Crown. Crown is the exposed part of the tooth. It projects above the gum. It lies outside the jawbone. Crown takes part in feeding activities. Covering of crown. Crown is externally covered by enamel. Enamel is thin, very hard, shiny white. Enamel is the hardest substance in the vertebrate body. Neck. Neck is the constricted region of the tooth. It lies between crown and root. It marks the junction of crown and root. Neck lies at the level of the gum margin. Root. Root is the basal part of the tooth. It is embedded in the jawbone. Root is lodged in a socket called alveolus. Root covering root is covered by a hard substance called cement or cementum. Cement is a modified bone tissue. Alveolus, tooth socket. Alveolus is a bony socket of the jawbone. It accommodates the root of the tooth. Alveolus provides firm attachment to the tooth. Periodontal membrane. The root is fixed to the alveolus by periodontal membrane. It is a vascular connective tissue layer. It is made of dense, fibrous connective tissue. Sharpies fibers. Periodontal membrane contains Sharpies fibers. These fibers run from cement to jawbone. They strengthen tooth attachment. Periodontal membrane allows slight movement of tooth. Composition of a tooth. A tooth is made of four main tissues: enamel, dentine cement pulp, enamel. Enamel covers the crown only. It is formed from epidermis. It is the hardest substance in vertebrates. It protects the tooth from wear. Enamel is absent on the root. Dentine. Dentine forms the main bulk of the tooth. It is a hard dermal bony substance. Chemically, dentine resembles bone. Distribution of dentine. In crown, dentine is covered by enamel. In root, dentine is covered by cement, pulp cavity. Inside the tooth is a narrow cavity called pulp cavity. It is filled with a soft connective tissue called pulp. Contents of pulp: blood vessels, nerves. Pulp provides nutrition to the tooth. Pulp maintains vitality of the tooth. Odontoblasts. Pulp cavity is lined by bone cells called odontoblasts. Odontoblasts secrete dentine. Odontoblastic processes. Odontoblasts send fine protoplasmic processes. These processes extend into canaliculi of dentine. They help in nourishment and sensitivity, root canal and apical opening. Pulp cavity extends into the root as a root canal. At the base of the root, an opening is present. This opening allows entry of blood vessels, entry of nerves, open-rooted and closed-rooted teeth. Open-rooted teeth. Pulp cavity remains open at the base. Teeth continue to grow throughout life, seen in rodents, lagomorphs, elephants. Such teeth are called open-rooted teeth. Closed-rooted teeth. Pulp cavity becomes closed at a certain age. Nutrition stops after closure. Further growth ceases, seen in majority of mammals, humans. Such teeth are called closed-rooted teeth. Gum, gingiva. Gum surrounds the base of the crown. It strengthens attachment of tooth. It protects the neck region of tooth. Important points: enamel is epidermal in origin. Dentine, cement and pulp are dermal in origin. Enamel is hardest substance in body. Odontoblasts line pulp cavity. Open-rooted teeth grow continuously. Closed-rooted teeth stop growing. Development of a tooth in mammals. General introduction. Teeth develop in the gum or soft tissue of jaws. Development occurs along borders of premaxillary, maxillary, dentaries. Tooth development is similar to placoid scale development. Teeth develop partly from epidermis and partly from dermis. Origin of tooth tissues. Enamel develops from epidermis, ectoderm, dentine, cement and pulp develop from dermis or mesenchyme. Formation of dental lamina. Tooth development begins with ectoderm thickening. Thickening occurs along margin of jawbone. Basal layer of ectoderm is called Malpighian layer. This layer grows inward into dermis. This inward growth forms a ridge-like structure. This structure is called dental lamina. Dental lamina retains connection with outer epidermis. Formation of tooth germs. Mesodermal cells multiply beneath dental lamina. They form solid bud-like outgrowths. These structures are called tooth germs. Tooth germs form at regular intervals. Number of tooth germs equals number of milk teeth. Formation of enamel organ. Each tooth germ develops a cup-shaped epithelial structure. This inverted cup is called enamel organ. Enamel organ is ectodermal in origin. It later secretes enamel. Formation of dental papilla. Beneath the enamel organ, mesenchymal cells aggregate. This mass is called dental papilla. Dental papilla is mesodermal in origin. It later forms dentine and pulp. Differentiation of cells. Odontoblasts, cells of dental papilla, differentiate into odontoblasts. Odontoblasts line the outer surface of dental papilla. They secrete dentine. Amyloblasts, inner epithelial cells of enamel organ, become ameloblasts. Ameloblasts secrete enamel. Enamel forms a cap over dentine. Enamel is deposited only on crown. Enamel is not deposited on root. Formation of pulp cavity. Dental papilla persists as pulp tissue. Central cavity enlarges to form pulp cavity. Blood vessels and nerves enter pulp cavity. Entry occurs through basal opening. Formation of root and cement. Root region does not receive enamel. Around root, a hard layer appears. This layer is called cement or cementum. Cement is a modified bone tissue. Cement helps in fixing tooth to jaw. Eruption of tooth, cutting of tooth. Initially, tooth remains embedded in gum. Tooth gradually moves upward. Tooth pierces overlying epidermis. This process is called eruption or cutting of tooth. Completion of tooth formation. Odontoblasts become inactive after full formation. Tooth attains final shape and size. In most mammals, O growth stops after root closure. Continuous growth in certain mammals. In rodents and lagomorphs, odontoblasts remain active throughout life. Pulp cavity remains open at base. Teeth continue to grow continuously. Such teeth are called open-rooted teeth, closed-rooted teeth. In majority of mammals, including humans, basal opening of pulp cavity closes, blood supply becomes limited. Further growth stops. Such teeth are called closed-rooted teeth. Evolution of molars in mammals. Introduction. Molars are complex cheek teeth of mammals. They show great variation in shape and structure. Evolution of molars has been a debated topic. Two main theories explain molar evolution: Cope-Osborn tritubercular theory, concrescence, Rose theory. Concrescence theory, Rose's theory. Basic concept. According to this theory, molars evolved by fusion. Two or more simple conical teeth fused together. Fusion formed complex molar teeth. Explanation. Early vertebrates possessed simple conical teeth. These cones developed separately. Later, adjacent cones fused together. Fusion produced triconodont teeth, hexaconodont teeth, polyconodont teeth. Evidence and limitations. Theory is based mainly on morphological observation. Embryological evidence is weak and incomplete. No clear developmental fusion is observed. Status of the theory. Concrescent theory is not accepted today. It is rejected due to lack of embryological support. Cope-Osborn theory, tritubercular theory. Basic concept. This is the most accepted theory proposed by Cope and Osborn. Molars evolved from a single reptilian cone tooth, primitive reptilian tooth. Early reptiles had simple conical teeth. Thus, teeth had a single cusp. They were suited for simple feeding. Formation of triconodont tooth. The primitive cone elongated anteriorly and posteriorly. Two additional cones developed on sides. These three cones arranged linearly. This condition is called triconodont, found in fossil Mesozoic mammals. Example: triconodon, formation of tritubercular tooth. Later, cones shifted in position. Three cusps arranged in triangular pattern. This stage is called tritubercular, considered a key stage in mammalian evolution, seen in fossil mammals like Spalacotherium. Further evolution of molars. Additional cusps developed from original cones. Cusps multiplied and rearranged. This resulted in hexaconodont teeth, polyconodont teeth. This led to wide variety of molars in mammals. Adaptive modification of molars. Molars modified according to feeding habits. Different cusp patterns evolved. Bonodont molars, rounded cusps, suitable for mixed diet, seen in humans and monkeys. Secodont molars, sharp cutting edges, suitable for flesh eating, seen in carnivores. Selenodont molars, crescent-shaped ridges, suitable for grinding plant material, seen in ruminants and horses. Lopodont molars, cusps form transverse ridges called lofts, suitable for coarse vegetation, seen in elephants and manatees. Importance of tritubercular stage. Tritubercular tooth is fundamental to mammals. Most modern mammalian molars evolved from it. Provides evolutionary link between reptiles and mammals. Significance of molar evolution. Explains diversity of mammalian dentition. Shows relationship between teeth and diet. Helps trace mammalian ancestry. Useful in paleontology and taxonomy. Important points. Molars evolved from single conical teeth. Tritubercular stage is crucial. Concrescence theory lacks embryological proof. Copos-Born theory is widely accepted. Final significance. Dentition reflects diet and habitat. Essential for taxonomy and evolution. Important for understanding mammalian biology